Okay, thank you all very much for joining this special symposium on stress resilience and digital health in the era of COVID-19. I don't think I have to remind anyone in the audience how special, to put it mildly, last year has been, and certainly in the area of New York and environments, and then later on in the rest of the country, and now, as we all know, in the rest of the world, especially in India and Brazil. So this has been a time when stress, and therefore its mirror image, resilience, has been extremely important in the effects it had on the healthcare providers, but also, of course, the general public and the patients. So we thought it would be topical to bring together a group of excellent speakers, all from the Mount Sinai School of Medicine, the Icahn School of Medicine here in New York, to present and share with you very novel data on these different aspects like stress, resilience, and digital health. And also we'll be closing with a special program that we developed here to actually help and support the healthcare providers and other people who have suffered physically and mentally from the COVID pandemic. So what you'll be hearing is how stress works in the brain and body, how we can facilitate resilience and recovery. And we'll cover that in 20 to 25-minute talks where we have preclinical and clinical stress research presented by Drs. Russo and Murrow in that order. The Warrior Watch results, which is a very novel way of actually monitoring stress and resilience by Dr. Fayyad. And then, like I said, what are we actually doing? How are we helping the healthcare workers? And that will be presented by Dr. Marin. So I think we'll be offering you a very broad view on stress, resilience, and digital health. Now, in your original program, you must have seen that Dr. Chiney would give a brief introduction. Unfortunately, he has apologized. He couldn't be present. So that's why I'm giving you a little bit more lengthy introduction on this topic. So we hope you're going to enjoy this. Like I said, it's novel data. I think it's a broad spectrum of aspects that we'll be covering. Like I said, also, I think it's extremely topical and relevant for everyone who has gone through this COVID pandemic and who still has got to deal with the sequelae. So with that, I'm happy to introduce Dr. Scott Russo, who will be the first speaker. He is director of the Mount Sinai Center for Affective Neuroscience. He received his PhD in psychology from the City University of New York in 2003, and then completed his postdoctoral work in psychiatry and psychology at the University of Texas Southwestern before joining us here in 2008. Dr. Russo is very well known for his contributions to understanding the neural and immunological basis of neuropsychiatric disorders. His translational studies have identified novel disease mechanisms in depressed humans that play causal roles in the expression of depression-like behaviors in rodent models. He has also identified novel circuitry in the brain that controls aberrant social behaviors, leading to new perspectives about social dysfunction in neuropsychiatric illness. So I'm very happy, Scott, to introduce you, to give you the floor, the virtual floor on your presentations, and please take it away. Thank you very much.
Thanks so much, René. It's really a pleasure to be here today. I'm going to share some fully unpublished work with you all today. So this is hot off the presses and not available anywhere but this Mount Sinai feed. As René mentioned, my lab is very interested, at least part of it is interested in identifying the role that certain brain circuits play in stress susceptibility and stress resilience. And I'm sure I don't have to remind people that last year has been a particularly stressful and trying times. I think probably about a year ago from today, we were all in this mode, thinking about where we might find our next roll of toilet paper. And that quickly then led to the realization that we were home with children and trying to work and maintain our labs, maintain our jobs. maintain some semblance of normalcy. It also was associated with significant social isolation. And then we learned, I think, early on in the pandemic that the virus itself, if you were unlucky enough to have contracted it, could have some long-lasting neuropsychiatric effects, and partly due, or at least it's thought, due to changes in the way in which the virus is acting on the immune system and how that affects your brain. But there's another type of stress that ramped up really significantly and in a very concerning way, and this is interpersonal violence. This is a perspective written in the New England Journal where they called it a pandemic within the pandemic. This was particularly difficult for women, but across the spectrum, we saw increases in interpersonal violence, probably because we were stuck in the confines of four walls together for very long periods of time and were stressed out. But just to show you some data, this is a study from the US showing rates of nearly, upwards of 10 to almost 30% increases in interpersonal violence that we saw last year. This wasn't isolated to the United States. Data out of Europe and South America show similar rates of increases, upwards of 25 to 30%. And what is this type of increase in interpersonal violence? What is this going to do to the mental health of our population? Well, it's not quite clear yet. I don't think we've completely rounded the corner on the pandemic, and so the numbers are starting to trickle in. We've certainly seen a significant rise in neuropsychiatric events. But if we go back to the literature and look at the impacts of such interpersonal violence, it's quite dramatic and concerning. So this is a study looking at the activity patterns in brain regions that control our responses to rewarding and pleasurable events. And they found that if you experienced relational victimization in the past year, that these brain regions weren't functioning as they were normally. And ultimately, it was impairing your ability to experience rewarding events in your life. Similar studies in children have shown that victimization or violence towards them, or even just childhood maltreatment in general, can lead to an increase in the child's reactivity to threatening stimuli, suggesting that they experience aversive and threatening stimuli in the environment with a far more robust response. So that led us to think about what this might mean. Our hypothesis going into these studies was that negative social experience, one that might be experienced through partner violence or aggression outside of your immediate personal relationships, may actually lead to a heightened activation of brain regions that are typically involved in our perception of stress or threat, and that activation of the overactivation or inappropriate activation of those stress circuits in times when they shouldn't be on may actually occlude our ability to experience rewarding events. And one in particular is social behavior in and of itself. So our ability to experience social behavior as rewarding and our desire to engage in social interaction. And that ultimately this may lead to severe social avoidance behavior. So to get at this, we use mouse models because it allows us to better interrogate neural circuit mechanisms to define functional circuits that may control such a response, but also to test novel targets, either at the drug level or at the neuromodulatory level, to determine circuits that we might tap into to promote resilience to such a social stress, thereby kind of reversing these maladaptive behavioral responses. So to mimic violence or aggression, we use a social defeat stress model. This is an established model of stress. We select large aggressive mice that are screened for for being particularly aggressive and their willingness to subordinate a broad range of social targets. We can do this in both males and females, but in both of our models, the aggressor is always a male mouse. So we then introduce a smaller, more subordinate C57 black 6 mouse into the cage with this aggressive bully. They are socially subordinated for 5 to 10 minutes each day over a 10-day period. This is a very chronic and consistent social stress that they experience. During the remainder of each day, they're housed on the other side of a plexiglass divider. So it's thought that they're experiencing both the physical stressor, but also a significant psychological stressor. We can then test their social behavior and determine whether they're susceptible, meaning they show a particular behavioral phenotype, or whether they're resilient and have managed to resist the development of any significant behavioral maladaptations. We do this first by screening social interaction, placing a novel CD1 mouse into a goal box here, and then allowing the experimental C57 mouse to interact with that mouse in a goal box. Mice that interact normally or similar to controls, and the quantification is down here, we term them resilient. Mice that avoid the social target when they're present in the cage, we call them susceptible. It's important to note that susceptibility and resilience are not black and white terms. You can be susceptible to certain features of the stress and resilient to others, and vice versa. But in general, we define susceptibility as a loss of social interaction that's associated with a host of physical abnormalities, including a type 2 diabetes-like effect and changes in the peripheral immune system indicative of chronic inflammation. We don't really know exactly what drives susceptibility and resilience. These experimental mice, it's important to note, are genetically identical. They're inbred strains, and so the difference between coping or susceptibility and resilience is not defined by some genetic code like it might be in humans, but rather we think it's due to either behavioral or other related phenomenon that either allow the animal to adapt in one way versus the other. And so one of the things that we've begun to look at is behavioral coping strategies. What is the experimental animal doing in the cage while they're interacting with this large aggressive CD1 mouse? We've identified 3 behavioral coping strategies, which we've termed passive, active, or outright fighting back. And these types of behaviors can be quantified during the social defeat episode. Passive coping is when the animal kind of gives up. It's maybe more akin to learned helplessness behavior. They throw their arms or their paws in the air and say, "I'm done, you win." Active coping is when the animal actively tries to avoid such social subordination, jumps and hops away from the aggressor and runs around the cages. And then fighting back is exactly what it sounds like. They put their dukes up and push the aggressor back and try to limit the amount of aggression that they experience. And in fact, if you quantify these coping scores over time, you can see that actually the degree to which the animal is socially avoidant correlates with coping, with the higher coping scores reflecting the more active approaches of running away and fighting back, and the lower coping scores reflective of these more passive responses. So it does seem, at least at some level, that it's a behavioral strategy. Now, what are the kind of brain circuit mechanisms or underlying mechanisms that drive this is something we're quite interested in. But I do want to highlight one thing. We've used this model over the years, and we've interpreted these reductions in social behavior as being a loss of social reward or an inability to experience social interaction as being pleasurable. But in reality, with these types of paradigms, I think it's really difficult to make those conclusions. And the reason is that, as I mentioned here, our output, our test is looking at interaction with this CD1 mouse. Now, regardless of the fact that it's not an aggressor that it's seen before, that it's novel, that it's never actually been attacked or subordinated by it, mice generalize cues. And it's very likely that the avoidance of this target is based on fear of the traumatic experience that it had with one mouse that looked very similar to this. So I don't think these types of behavioral assays really tap into this idea or this construct of social reward and anhedonia. So to do that, we've established a slightly different set of derivations to the model. We still do the chronic social defeat stress the same way we've always done it. We still define susceptibility and resilience as the interaction time spent with this novel CD1 mouse, but we now test social behavior in the presence of a non-threatening, same strain, same sex juvenile mouse. This is a 3 to 4 week old pup, so non-threatening in any way, shape, or form. Under control conditions, 3-week-old pups are typically rewarding to adult male and female mice. And so we think that by looking into the types of interactions that occur under these circumstances, we may better tap into social reward processes. So in fact, when we do this new test of juvenile social interaction, we still see that susceptible mice show reduced investigation time with the juvenile, and they show more bouts of active avoidance. Active avoidance is defined when the juvenile approaches the adult mouse and the adult mouse retracts and runs away. So this does seem to impair social behavior even to non-threatening social stimuli. But to get at reward preference, we use a slightly different model. This was borrowed from the drug addiction literature. It's a conditioned place preference apparatus that's been used historically to measure the rewarding properties of all drugs of abuse. In the model, we have contextually distinct chambers defined either by these white and black stripes or gray zones. The contexts differ by tactile cues as well as visual cues. This is really so that the animal can determine and learn environments associated with a certain cue. In the case of drug addiction, what we would often do is inject a rewarding dose of a drug of abuse into one of the two chambers, condition the animal 30 minutes a day for 4 days, and then by the end of that 4 days, you put the animal back into the box, allowing them free access to both conditioned chambers, and you test to see where their preference is. And for all addictive drugs of abuse, their preference is toward the side in which they receive the injection of the rewarding dose of the drug of abuse. We replace the drug of abuse in our model with a juvenile, same sex, same strain mouse, and we do the conditioning in a similar fashion. And then at the end of that conditioning session, we again place the animals back into the box and we look to see which side of the chambers they like to spend their time in. Both male and female mice spend more time after training in the side in which they were allowed to interact with that juvenile male mouse, and that's shown in these red dots up here on both sides. So male and female adult mice, this is under control conditions, both find these juvenile C57 mice to be rewarding. Following social defeat stress, we see a different pattern. So again, we repeated the social preference that we observe in control mice. These are mice that did not experience any significant social defeat stress. Same phenotype is observed in resilient mice, but susceptible mice fail to develop this conditioned reward response, and they do not distinguish between the social paired chamber and the social unpaired chamber. They don't find it aversive either, and I think that's an important point. For example, if you were to put an animal into one of these chambers with a rewarding dose of opiates and then hit them with an opiate antagonist, they don't just prevent reward to that opiate, they actually produce aversion because it's an aversive internal state. So we're not seeing an aversive internal state, but what we're seeing is a blockade of the ability of these animals to develop conditioned reward to these social targets. So to get at the brain mechanisms, we took a more high altitude approach to start with. We wanted to get a general sense of what brain areas were becoming activated in response to a juvenile social target in control susceptible and resilient mice following the 10-day social defeat stress paradigm. So we used a brain clearing procedure that allows us to clear the brain tissue out so that it's fairly transparent, and you can then stain for markers of cellular activation throughout the entire extent of the brain tissue. In this case, we use CFOS, which is an immediate early gene that's activated in response to neural activity. So it's a good broad marker to look at neural activity in response to a particular stimuli. We sacrificed mice following, as I said, 10 days of social defeat stress and a juvenile social interaction. So we're really looking at CFOS that's being induced in response to that juvenile social interaction. And what you can see as you look three-dimensionally through the tissue sections, you can see nice staining of CFOS. As we zoom in, you'll be able to see it better, but these are small circular dots that you'll see throughout, and each one of those is associated with a specific cell type in the brain, and then it allows us to kind of quantify the number of those cells that we see in various brain regions and throughout. Okay, so this is a table just showing the brain regions that came up. You can see entorhinal cortex came up as strongly activated in susceptible mice compared to control and resilient, the lateral septum, and a whole host of other structures. This table is from male mice. The male and female brain circuits that were activated in response to this are quite different, but we did note that the lateral septum, which was one of our top regulated regions, was among the very few that were similarly regulated in males and females. Given that we saw similar behavioral phenotypes, we wanted to explore the lateral septum and its target regions in controlling some of these maladaptive responses to social stress. So we found within the lateral septum that a specific type of cell that expresses the neuropeptide neurotensin and then also the inhibitory neurotransmitter GABA was the cell type that was predominantly activated in response to these social targets. So in the left-hand panel, I'm showing you in situ hybridization images in control, resilient, and susceptible mice. Again, this is following 10 days of social defeat and a juvenile social interaction test. We normalize these data to animals that go through 10 days of social defeat without the juvenile interaction test. So we're measuring CFOS that's being activated by that juvenile social interaction. CFOS is in red, neurotensin is in green, and you can see that susceptible mice have strong activation based on CFOS expression within these neurotensin cells in the lateral septum. And we see similar responses in male and female rodents. Resilient mice, though going through the stress, do not seem to activate these cell types in response to the interaction. So we wanted to dig a little bit deeper into this cell type. There wasn't a lot known with relation to stress. The lateral septum itself is really thought to play an integral role in interfacing between, for example, memory circuits and reward circuits, and has been shown to play roles in the response to reward, but we didn't know anything about these LSNT neurons. So interestingly, so the way that we did this was to use an in vivo approach so that we could look at neural activity in real time as these mice were interacting either with a social target, a stressful event, or some other stimuli with a specific valence tag to it. So we used the calcium indicator GCAMP6. We did this by injecting an adeno-associated virus that was Cre-dependent, meaning that it only expresses in cells that also express the enzyme Cre-recombinase. We did this into an animal that was engineered to only express Cre-recombinase in those neurotensin cells, which means that we can now measure activity, calcium activity, specifically within the neurotensin cells in the lateral septum. Calcium activity is a surrogate marker of neuronal action potentials, as it's required for all nerve cells in the brain to fire. So that by measuring changes in calcium activity during this social interaction, we can now hypothesize or conclude the activity patterns that are associated with certain behavioral events. So we put the animals through chronic social defeat stress, we test for susceptibility and resilience, we then measure activity of LSNT neurons during juvenile social interaction. And you can see that these cells don't respond in control mice to the juvenile. Now, this is a rewarding event for these animals, and so it does kind of argue that these cells are not necessarily playing a role in sensing reward or responding to rewards in the environment, at least in unstressed control mice. In resilient and susceptible mice, we see a pattern that's quite different from one another. Resilient mice, again, look very much like controls. These cells do neither activate nor inhibit in response to a juvenile social target. But susceptible mice have a very strong activation each time they interact. These red ticks at the top reflect social bouts with the juvenile. And you can see that the calcium activity goes way up in response to juvenile interactions. And this is just the quantification of the total amount of fluorescence emitted by the calcium indicator over the entire session of social interactions. So it does appear that these cells might be sensing something more akin to threat or stress, but we're not quite sure at this point. So in order to get at that question a little bit more, or to examine that question a little bit more closely, we wanted to remove the social context and add in non-social threats or stressors and see how these neurons respond. So we did two different ones. One actually was a social threat, the other was a non-social threat. The social threat was a CD1 aggressor attacking it, and you can see that these LSNT neurons respond very robustly to attack signals, almost though similar to a juvenile, which is quite shocking that these cells now activate to levels comparable to when they're being attacked by a CD1 mouse. So that tells you how stressful the juvenile social interaction must be, or just maybe general interaction. We also did an additional stress where we hung the animals by their tails, and you can again see very strong activation of these circuits. Interestingly, rewarding stimuli do not activate or inhibit. So again, these cells seem to be coming on in response to threat or stress. To determine the functional relevance of these cells in terms of modifying this behavior, we used a chemogenetic approach. This is a synthetic receptor that responds only to a synthetic drug. And so this allows us to either activate or inhibit these cells. I'll just show you how we did this. We chose previously susceptible mice, and we then inhibited these LSNT neurons. And by doing so, we can bring social interaction way up, comparable to those that we see in resilient mice. And we can then promote a more normal social CPP. We can take previously resilient mice and do the opposite, where we can now promote social avoidance by activating the LSNT neurons and then occluding social reward. I think I'm going to skip this last part in the interest of time. I'll just say with this that we're now starting to probe circuits downstream or upstream of the lateral septum neurotensin cells to see how they may be occluding social reward. Our hypothesis is that these cells are projecting to primary reward centers like the nucleus accumbens and nucleus of the diagonal band, and that by direct communication, they're promoting or opposing social reward behaviors. So they're occluding social reward in the context of susceptible mice. And with that, I will just thank people in my lab that did the work. In particular, I'd like to thank Long Lee. This work was led by Long, Catherine, Roman, Joey, and Tony, and the rest of my lab and our funding sources, of course. And thank you all for your attention. I'm happy to answer questions.
Thank you very much, Scott. That was a great, very original, great studies. You're right. We have time for one or two questions, and people can use the Q&A box, which should at the bottom of your screen. And then you can type in the question, and Scott Russo will then read and answer the question. So please go ahead with the Q&A. In the meantime, Scott, I do have a question. What do you think is the main characteristic between the resilient and the susceptible mice? I mean, if you would put your money, they're all genetically identical, I think, so that's a little bit difficult. But I mean, where do you think is the main part of the answer?
I certainly think there's developmental issues that we don't control for. that we can't really define coming into it. So there's certainly, neurobiological differences based on how they develop. At a behavioral level, we kind of think about it as them taking a particular strategy. Individuals are faced with decisions all the time, and you have to choose what your strategy is going to be in life. And I think it can be learned. In fact, the coping that I showed you early on is a learned event. We don't see those differences at the early stages of defeat. So I think maybe it's serendipitous. Maybe an organism just decides how to respond in real time during a threatening event. And if they respond one way, they learn to then continue to respond in that way. But again, in humans, it's quite more complicated, right? I think there's going to be genetic factors, developmental factors, and others. But
yeah. So Scott, in the meantime, we have two questions in the Q&A. If you, or actually three, if you can address them,
Yep.
Because we'd like to have them all three answered pretty briefly.
Sure.
That would be great.
So Lana wants to know about the coping strategies. They reminded her of behaviors that they observe in humans associated with attachment styles. Wondering how you think of early life experience. I mean, it's a fantastic question. What we can do in a controlled fashion is change early life experience. So we can change the maternal relationship, periods of maternal separation, for example, enhance these processes, so they promote resilience. So I think much like humans, early experience is going to be critical. And we're starting to kind of think about that in the context of reward as well, although we don't have a final answer. The second question is similar, and it's related more to the epigenetic landscape. Again, we haven't looked at the epigenetic landscape of the lateral septum NT neurons. My guess is that we will find broad differences between susceptible and resilient based upon published work from Eric Nestler's lab across other brain circuits. And again, I think this will be defined a lot by those early life experiences, that the epigenetic switch is determined by those early experiences, and they then change how you respond as an adult. And the last question is related to neuromodulators in stress resilience, for example, dopamine and serotonin. It's very complicated. I'll speak just to dopamine because I think in the interest of time, we probably should move to the next speaker. And in fact, it's a great segue to the next speaker, who's going to be talking about testing novel therapeutics that are based on altering the physiology of dopamine neurons. And the work was conceived at the preclinical level using the social defeat stress model. Dopamine is complicated. It's actually increased firing of dopamine neurons is associated with increased stress susceptibility. And if you can silence those dopamine cells from firing to the extent that they do, it actually promotes resilience. So it's food for thought. I don't think it's through a dopamine specific mechanism. I think there's other mechanisms involved, other neuromodulators in dopamine cells. But James is going to really, I think, dig into that in the next talk in the context of human depression. So stay tuned.
Yeah, so Scott, I mean, there's one last question that you can just answer with yes or no. So that's a very easy one. Can you see it?
Oh, yeah, sure. Like drugs, are mice capable of experiencing extinction to social fear? Yes, and actually the susceptible mice don't extinguish as quickly or as readily. So.
All right, great. Scott, thanks again. Thanks so very much. And great, we could address all the questions. Thanks. And indeed, the next speaker is Dr. James Murrow. He's a psychiatrist, and he is the director of the Mount Sinai Depression and Anxiety Center for Discovery and Treatment. He completed his residency here at Mount Sinai and also a research fellowship in experimental therapeutics and clinical neuroscience, focusing on mood disorders at Mount Sinai. He also has a PhD in clinical research methodology and biostatistics, also here from Mount Sinai. He conducts clinical and translational research aimed at understanding the biological basis of mood and anxiety disorders in order to point the way towards new, more effective treatments. He was actively involved in the research that led to esketamine's FDA approval and has been breaking new ground with ezogabegib and is exploring the therapeutic potential of neuropeptide Y. In addition to his research activities, Dr. Murrow directs Mount Sinai treatment-resistant depression program, and he's really building up and leading his own group of researchers with which he's very successful. So James, thanks very much for joining us, and please take it away.
Thank you, Dr. Kahn, for that very generous introduction, and really a pleasure to be part of this important and very interesting symposium. And Scott, that was really, really elegant and inspiring work. So thank you. So in the next 20 minutes or so, I want to sort of pivot from what Scott was talking about and really continue that line of thought and sort of bring it into the clinic, into patients, which is really what our lab has focused on, and the Depression Center. The title of my talk is Discovering New Targets for Treating Stress Disorders. And in particular, I want to tell you about two targets and two sort of brief stories that hopefully are just the beginning chapters of actually moving towards identifying novel treatments. One story has to do with something called KCNQ channels, another with neuropeptide Y. These are my disclosures. There is IP filed around both neuropeptide Y and KCNQ channels. It's not commercialized or licensed. There's no income, but this is IP that's related to two of these things we'll be talking about today. Then the rest of the disclosure has to do with an institutional conflict related to ketamine, but I won't be talking about ketamine today. So this is the outline of my talk, and let me kind of just jump right in. So let me start actually with depression and just kind of start with this, sort of laying the foundation. This has been referred to as the world's greatest health problem. This accounts for more years lived with disability around the world than essentially any other medical or psychiatric disorder. This is, of course, before COVID, depression is the most common outcome of severe or chronic stress, continuing from the discussion you heard in the prior talk. Just very briefly, what you see on the left is this is actually, this report is limited to looking at disability levels of all brain-based disorders. So this is all psychiatric, neurological substance, where the years lived with disability, that's a metric of disability at the population level, is shown on the y-axis. And so here bigger numbers are worse. And you can see here in this blue line, this is by age, years lived with disability. This is depression in light blue, or depressive disorders that dwarfs everything else. This is it's hard to, I think, emphasize the global health burden of depression. Most patients who die by suicide suffer from depression, and we need to do better with treatments. There have been substantial recent advances in treatments for depression, but we have a long way to go. I will just make a note. I got this metric from a report in Nature from a few years ago on the bottom right. There is a lot of discussion. If you look at the disability and the public health burden of something like depression, and then the funding for biomedical research, it's really quite small. It's about 1/10 of the investment, for example, in cancer, another major driver of disability. That's from fiscal year 2013. But in any event, COVID stress, we are extremely, of course, concerned. And as Scott mentioned, I think pretty early on in the pandemic, as people started realizing what was happening, I can still remember when we shut the labs down in March of last year. I think I remember saying to the group that we thought we would be back in about 2 weeks. So I think it's hard to overemphasize the level of uncertainty, the way all of our lives have been disrupted. And for those people that were most vulnerable to things like anxiety and depression, that already had chronic stress in their lives, that had less resources, those have been the individuals that have suffered the most. So there's a tight link here between COVID, psychological stress, and depression. This is just a little bit of data that many of you may have seen. This is from a report in Nature from a few months ago. For example, just looking at the US population based on a survey during the pandemic, you see down here, bottom right, 44% of individuals were reporting symptoms of anxiety or depression, compared to about 11% around the same time the year before. So we're talking about a 4-fold increase. So this is extremely concerning. I think we don't fully know what the effects of this essentially second pandemic of psychological stress will be. Just a little sort of med student primer. Most of you guys know this, but you know, so what is the effect? What's the link between stress and mental health? Obviously, that's a huge topic, but just to remind you, you know, we know that stress in and of itself is not necessarily bad, right? So what you're seeing is this classic curve on the right, where actually low to medium levels of stress, context appropriate stress actually improve performance. But then at some point, the stress increases either in its severity or chronicity, and performance starts going down. And again, symptoms of anxiety and depression are sort of the classic outcomes of that. And we can think about sort of, this was adapted from the late Bruce McEwen, a review by him. His lab did so much in this area to understand just the biology of stress, but we're talking about everything from sort of chronic low grade and then to sort of the severe or life-threatening stress that we think of as more classically associated with something like, right, PTSD, post-traumatic stress disorder. And just to come back locally, what was happening in March, April, May of 2020 in New York City. So I went back to the Times and I grabbed this data. The New York Times was tracking this closely. This is case numbers, hospitalizations, and death rates for New York City. And what you can see, peaking there about April 10th, we were averaging about 10,000 new cases a day. You can see the shape of the hospitalization rate around that time, April, and then this dramatic spike in deaths. And as you all know, the frontline healthcare workers bore, I would say, a unique brunt of this, in both simultaneously fearing for their own health and their families. Again, remember all the uncertainty. We didn't really understand how it was spread, what happens. So it was a very, very scary time, still is. But we know that the frontline healthcare workers are in particular a position of vulnerability, but of course, more broadly, the whole population suffering unprecedented stress related to COVID-19 and all the things like social isolation that came with the response to the pandemic. So let me shift now to drill down on this target I want to tell you about today, just briefly for context. So what are the illnesses we're talking about? We're talking about depression, anxiety, and PTSD. Essentially almost all medicines for these disorders, one way or another, increase either dopamine, serotonin, norepinephrine in the brain. So this is just a schematic of the serotonin neurons. These medicines work for many, many people, fortunately. They were discovered many decades ago through serendipity, and it's still the mainstay of medication treatment for these illnesses that complements the other primary form of treatment, which of course is psychotherapy and behavioral treatment. But there are problems, including a multi-week or month lag from the time someone starts the treatment until the time they actually derive benefit. This is a high-risk time, a time of risk of suicide in more severe individuals. And unfortunately, a significant minority of individuals actually will not derive meaningful benefit from these medicines. This is something I borrowed again from a Nature article some years ago, just sort of trying to take a step back. What are the different ways we can find new treatments for these disorders? So what you see here in the top panel, this is kind of the classic, sometimes called reverse translation. This is actually how we got to the majority of the treatments we have today. We observe an effect in humans. We sort of understand how it works in the brain, and then we can embark on, for example, the modern class of serotonin selective agents, were sort of engineered improvements in terms of side effects and things like that. But it really wasn't based on fundamental insights from neuroscience that you got a taste of, for example, with Scott's talk in the last section. And hopefully what I want to do in the remainder of my time here is give you an example of how we're trying to think about this, and really from the perspective of what are the fundamental effects of stress on brain circuits? How do we then measure that in individuals and people, and then use that as a platform to find new treatments? So fortunately for us, all of us, Scott did a good job of introducing us to this essential stress model in mice. This is the chronic social defeat model, and the basic insights that led to the clinical trial I'm going to tell you about really came out of Ming-Hu Han's lab, and many others such as Scott and Eric Nessler's lab have used this model to really understand what's going on in the brain and behaviorally in mice who may show what might be called a defeated or susceptible phenotype, or depressed versus resilient. And just to recall, there's an experimental mice that's exposed to a large aggressor day after day for 10 days. And then we can measure the multiple outcomes in the animal, such as social avoidance, that's time, social interaction test, and some other things that measure anhedonia, which is reduced response to pleasurable stimuli, and anxiety. And I want to drill down and tell you about just one sort of sliver of the biology of this as I've come to understand it. This is data that goes back to, from Eric's lab, from Krishnan et al. In Cell 2007. So over here again, you see, so there was a question before about dopamine neurons and the reward circuit. The story's a bit complicated, but suffice to say, for these purposes, following social defeat, right, the susceptible or sort of pro-depressant animals show an abnormal hyperactivity in these neurons, whereas the resilient or unsusceptible show essentially normal physiologic activity of these D2 projecting neurons from a fundamental a part in the brain, the ventral tegmental area, to this area that we've been hearing about, the nucleus accumbens, that you'll see me also refer to as the ventral striatum. And this shows this picture. And of course, a lot of work has been done at the epigenetic level and protein level and the functional level, understanding, well, why is it that, and how is it that resilient mice maintain a normal reward circuit, whereas the defeated or susceptible, depressed animals don't? And one thing that seems to happen is that the resilient mice have an upregulation in a particular family of potassium channels, which, if you remember, potassium channels signaling through there will tend to quiet down nerve cells, make them fire less. And so the theory is that this maintains in the resilient animals or unsusceptible relatively normal firing rates by virtue of more potassium channels compared to the susceptible. Okay, so why am I telling you all this? The insight from Minghoo Han was that, okay, so if you took, we know that in so-called resilient mice, they have, let's call it an upregulation or increased functioning of the specific KCNQ3 channel, or what's called a 2-3 channel, because the 2 and the 3 heterodimerize. But what if you could give a drug to the mice that actually were susceptible to the stress that could mimic that? These are drugs that are called positive allosteric modulators, or KCNQ openers. They would bind to the channel and essentially make the channels that are there do more. And so this is work by Alison Friedman from Minghoo's lab. And just very briefly, this is exactly what they found, that across multiple different KCNQ channel openers, they could actually reverse a depressive phenotype in the susceptible mice. So this is just an example. Here, the drug is labeled retigabine. That's the same drug as ezogabine. It's known as retigabine in Europe. Scott told you about the target mouse, that a susceptible mouse will show an abnormal behavior of sort of hunkering down in the corner rather than normally exploring a conspecific non-aggressive target. With treatment here, you show this is a normalization of behavior. Same story in this classic, what's called forced swim test, where treatment with the KCNQ opener, retigabine or ezogabine, it shows an improvement in time spent immobile, which has been associated with antidepressant effects in people. On the right, this is an image used by courtesy from Mark Kaplan that very briefly just tries to basically boil down 6 years of research into the basic concept that if we can increase KCNQ function in brain, that may be protective against stress, it may be antidepressant. We can do that in people, and we can use a technique of functional neuroimaging to actually look and see, is this working through its effects on the same brain region, the nucleus accumbens or ventral striatum. And so that's what we did. We did a pilot study, but this is work through funding by the National Institute of Mental Health. This was just published in the American Journal of Psychiatry. I think it's ahead of print now. This is a proof of concept, the first randomized controlled trial this is in adults with major depression, 45 individuals that had depression, elevated anhedonia, and they were treated with this KCNQ opener, ezogabine, or placebo. The primary endpoint was actually change from baseline over the 5-week treatment in the ventral striatum during a reward task, an adaptation of the classic monetary incentive delay task. And then secondarily, we looked at measures of depression, primarily the MADRS, which is a common clinical measure of depression, as well as measures of anhedonia. So what did we find? These are the results. On the left, you see a schematic of this version of the monetary incentive delay task that we implemented with collaboration with Emily Stern. I won't go through the details, but essentially, while the person's in the scanner, you can isolate brain activity that's during the conditions where the individual's told they could get a reward in the form of money if they perform correctly. You can aggregate all that on average, look at how people do at baseline before the drug and then after. And what you see on the right is the region of the human ventral striatum, which corresponds to the nucleus accumbens in rodents. That's been such a focus of the relevant circuitry for these behaviors, such as depression and anhedonia. Our hypothesis was that under treatment, activity should go up in this region. And what I didn't show you data on, but there's data that, as you might expect, patients with depression had abnormal activation or reduced activation of the system to reward. So we hypothesized that the activity would be increased. And in fact, this is what we saw numerically, although there was a lot of variation. This actually did not reach, this just missed statistical significance, but it was in line with our hypothesis that under treatment with ezogabine, at the end of the trial compared to baseline, there was an increase, and under placebo, there was essentially no change. Perhaps most surprisingly, we saw a very large effect on the clinical measures of depression and anhedonia. I'm showing you the depression measures here. On the left is the MADRS score here. This is change from baseline, where negative numbers are good. That's the number of points you've improved. In general, a 10-point improvement is considered a fairly good benchmark. By the end of the trial, these individuals were exceeding 15 points better if they were treated with ezogabine versus about 10 with placebo. And this is a relatively large difference. And we saw similar measures with self-reports of depression, which is shown on the right. So this was very exciting. And this work forms the basis of a second trial we'll be doing with the support of the NIMH to explore this further. We simply put forward a very simplistic hypothesis based on this work, essentially that we might call it the KCNQ channel hypothesis of depression, and basically that at least some patients with depression have deficient signaling for one reason or another at these KCNQ2-3 channels. It might be specific to the reward system. We don't know that yet. And the corollary, of course, is that enhancing signaling at these channels will improve symptoms of depression, perhaps specifically anhedonia. I know I went through that fast, but we don't have a lot of time, and I want to tell you about one more target. So I want to shift gears, telling you about our work with neuropeptide Y. Here, just on this slide, I want to pivot back to more of the classic stress or trauma of that we would consider, for example, something like PTSD. It turns out that neuropeptide Y is a circulating hormone which has been observed to be low in individuals that have PTSD. We're not sure if it was low before, but it's hypothesized to be a pro-resilient hormone. For example, work by Dennis Charney and others, I'm not showing here, but had shown that soldiers under stress, the greater their NPY level in the blood went up, the better they did under simulated interrogation. Here I'm showing you a classic study in the cerebrospinal fluid. Patients with PTSD have low NPY, okay, compared to healthy subjects. And there's a very large evidence base that essentially NPY is anti-anxiety in stress, basic stress models. And some of the regions that you heard about in the last talk, but are very important when we think about regions that register threat and fear. We're talking about, for example, the amygdala and its connections with the frontal cortex. These are regions that express relatively high levels of NPY. And again, it can be shown that, for example, a direct injection of NPY receptor Y1 agonists can block the expression of fear or reduce fear learning. On this slide, I'm just showing you distribution of the NPY receptors in rodent brain. Again, showing some of these key regions that we care about. Like I said, I don't have time to tell you the whole story, but at Mount Sinai, we've been working on trying to figure out if we can get neuropeptide Y into brain, whether there's any signal that it could be effective for a disorder like PTSD for greater than 10 years. And there's a long story. There's many CNS conditions where we would like to figure out how to get peptides into brain, but it's not easy. They're broken down in the stomach. They don't go through the blood-brain barrier easily. There is something called the direct nose to brain route. And this is what we utilized for an initial study we did in PTSD, where this here, the artist is showing examples of oxytocin, similar problem. Oxytocin as a receptor, as a peptide, wanting to get that into brain, but the same concept applies for neuropeptide Y. So we partner with a company that develops a specific device to try to get particles up into the very top of the nasal cavity where they can actually have direct communication and migrate into the brain. And it's been shown in rodents can actually get into critical limbic structures like amygdala and hippocampus. NPY can when delivered in rodents through this direct nose to brain route. So just in my last 2 or 3 slides, I wanted to share with you a study that was published in the last few years of a randomized controlled dose ranging study in patients with PTSD. We had no idea, this had never been done before. We didn't know how much of this would get into the brain, what dose would be needed, but there was basic rodent work where we knew what dose would block stress in rodents, and then we could convert that to humans. And we ended up with a range here from all the way from giving 1.4 milligrams to 6.9. And individuals came in, were randomized to one dose or the other, all the way up to the highest dose. And the primary goal was to see was this safe. But we also looked for anxiety signals. And the way we did that was each individual came in on 2 separate treatment days, that's shown in panel A, and they underwent what's called a stress provocation procedure that I won't get into the details, but essentially it's a trauma script that's developed by the patient and the clinician ahead of time and then played back to them on the treatment day. And this reliably provokes symptoms of anxiety. And we treated them 30 minutes before with either NPY or placebo. This was a double-blind crossover study shown in A, and you see the timeline of, for example, the treatment at time 0, trauma script at 30 minutes. It takes about 1.5 minutes, and then we start getting symptoms throughout the rest of the day. So what did we find? First, we found that NPY was well tolerated up to the highest dose. We don't know how high we need to go, but we had a hypothesis just in terms of the efficacy that higher doses should show a greater benefit than lower doses on anxiety. And that's actually what we found. This graph basically shows the difference between NPY and placebo on one measure of anxiety that was significant, the Beck Anxiety Inventory. Here, more negative means there's more of a benefit of NPY over placebo. And if you fit a line, what you end up finding is that at the high doses, but not the low, there's a significant blunting of anxiety of NPY compared to placebo. Very preliminary. I should point out, there's only a few patients at each dose. This is not different than 0 at any individual dose, but there's a trend here. So we were excited. This was the first proof of principle that it was possible that NPY could be having an anti-anxiety signal in patients. This is my last data slide. This was just published. This is work led by Alexander Mate at the Karolinska Institute that we had the pleasure of collaborating on and supporting his research through collaborations, sharing our IND and other things. All the patients here were enrolled in his lab. He was interested in whether, very similar approach, used the same device, same manufacturer of neuropeptide Y. In this case, it was 6.8 milligrams. These are in patients with depression, and asked the simple question, was there a trend of reduced MADRS score within the first 1 or 2 days after treatment? What I'm showing you here in this graph is this is change in MADRS score. So now, the bigger the number, the bigger the improvement. Okay, this is +1 hour, 5, 24 hours, and 48 under NPY and placebo. What you see here is a trend over 24 hours and a statistically significant effect of NPY over placebo 1 day after treatment. By 48 hours, there actually was no difference. It should be pointed out that per protocol, the 48 hour is actually the primary outcome. So again, highly preliminary. These were 12 individuals treated with NPY, 18 under placebo, but another study in a different stress disorder in another country. So this gives us some hope that we really may have some traction with pursuing neuropeptide Y as a target. So let me just wrap up. So what I've told you about today, and again, this is well known, but we're very concerned that COVID is going to have far-reaching consequences in terms of the effect of stress on mental health. And these, what I'm calling stress disorders, depression, anxiety, PTSD, are already a major cause of suffering and disability. And I'm trying to, and the flow, I think, from Scott's to mine, and you'll hear the other talks, my pitch to you all is that we need new approaches that are based on neuroscience and understand the basic biology to really move the field forward. I've given you a little bit of data, and it really just examples that it could be that enhancing signaling at KCNQ channels within the reward circuit may be pro-resilient, may be antidepressant. I've shown with you a little bit of data that enhancing NPY signaling within the fear circuit may also be pro-resilient and anti-anxiety or anti-depression, and that future research really must endeavor to translate insights from neuroscience into actionable therapeutics for patients. So really enjoyed being able to present this work to you, and be happy to take questions.
Thanks very much, James. That was a great talk, and you know, super that this really looks that, you know, we may be having new drugs if you can get them in the brain, but you're working very hard for that. So actually there are 3 questions in the Q&A, if you can see them. I mean, there are 4, but the first one is just saying thank you.
Sure, okay.
Which is very nice. But if you can start with the one from Jill and work your way down.
Sure. Question from Jill: can NPY be measured in the blood or urine? It can be measured in the blood, and in fact, it is found. It does circulate in blood. The problem is that we don't think that NPY from the blood is really getting into brain because it doesn't cross the blood-brain barrier. So the typical metrics of, for example, you might want to know, you know, in that study, what was the blood level achieved NPY? It turns out there's really not much of a correlation. It wouldn't be helpful because we don't think the drug is going from blood into brain. Urine, I'm not entirely sure about that. Actually, I don't think so. Okay. Next question from Ann Kush: does KCNQ-based treatment target only VTA? Right. Okay, so I alluded to this a little bit. So when we give the drug, it goes everywhere in the brain. So what we don't know is where in the brain is its true mechanism of action. I can tell you, and the reason why we focused in on our measurement of the reward circuit in humans is because the work from Ming-Hu Han's lab, before they moved to the data I showed you, I may not have mentioned this, but they systemically injected ezogabine or retigabine or other drugs into the mouse to look at the behavioral treatment effects, because that was important for translation. But they've done a bunch of work showing that actually it is direct injection. They can directly inject the drug into VTA, into the circuit, and show that it's working in that system through those receptors. But there's a lot more we have to know about how exactly is it working in brain. This is sort of the issue sometimes described as off-target effects that we're always dealing with in medicine and psychopharmacology. Question from Alyssa: could one's exposure to early stress be impacting? So the answer is absolutely.
Okay, I mean, James, I think you have to really read it out loud, because otherwise people.
Oh, I'm sorry. Oh, I'm sorry. I forgot. Now I'm the only one that can read it. I'm sorry. Thank you. So yes, the question is, could one's exposure to early life stress be impacting the efficacy of current common pharmacologic interventions used for anxiety or depression in those individuals? So we think absolutely it can, and there's quite a bit of data that individuals that have suffered early life stress or maltreatment respond less favorably to commonly used medicines like SSRIs. So we absolutely. Good
All right, James. Thanks so very much. Again,
okay, thank you.
Very inspiring talk with, clearly clinical implications. And then we move on to the next speaker, Dr. Zahi Fayyad, which is also going to be, I think, very interesting, very novel, and showing from a totally different angle what we can do to, help people cope with stress and increase their resilience. So the next speaker is Dr. Zahi Fayyad. He is the founding director of Mount Sinai's Biomedical Engineering and Imaging Institute. He received his MS in Engineering, a Master of Science in Engineering from Johns Hopkins, and his PhD from Penn. His interdisciplinary research, from engineering to biology and from preclinical to clinical investigations, has been dedicated to the detection and prevention of cardiovascular research, with many seminal contributions in the field of multimodality, biochemical imaging, and nanomedicine. His work is recently expanding in understanding the effect of stress on the immune system and cardiovascular disease, as well as the use of mobile technologies for disease detection and management. And on the latter, he's going to present. And we're very glad that Dr. Fayyad is also now interested in stress and mental disorders, because we'd love to collaborate with him. He is a great collaborator and a great scientist and a great leader as well. So Zahi, thanks so much for joining us, and we're looking very much forward to your talk.
Thank you, René, and I really appreciate all the mentors that I'm having at Mount Sinai and collaborators to help me better understand that the brain and heart and immune system connection. So this is one of the projects, part of my collaboration on this area that really spun off during the pandemic, and it was a great way to connect with people that I didn't know, other faculty members who have common interests, but looking at it from a different way. And the other thing too, that the COVID and the pandemic has pushed us to adopt new technology that is touchless, where you could remotely collect data on your subject because of the pandemic, but also taught us that this also can, and in the future, translate even further. So I'm going to talk to you about this work that we call the Warrior Watch, and I will explain to you what that is. And this is using that Apple Watch to measure stress and resilience in healthcare workers. And I'm also going to cover to you how we started this program in terms of the COVID and the infection leading to this work. So I don't need to motivate you too much about this. A lot continues to be written and discovered in terms of the impact of COVID and the COVID infection on people, so and especially also on the vulnerable population, and one of them is the healthcare workers. So these healthcare workers have a threefold increased risk of COVID infection because of the work they do, and 35 of people are asymptomatic carrier but are contagious. So there's the need, obviously, to try to detect this disease differently. The other aspect, and this is what we're talking about, and this really is the work by my colleague to try to understand the psychological impact of COVID. But in this case, I'm going to talk to you about our work looking at healthcare workers, where they have been a lot discussed. And as James mentioned, this is going to remain for a very long time with us, the aspect of depression, anxiety, insomnia, and distress. So mobile health or mobile technologies is on the rise, and it's here to stay. And we have seen the impact of telehealth during the pandemic, but also the adoption of multiple tools, technologically based, mobile based, and it's predicting that this is going to have and continues to have an impact both from a patient or subject research and clinical delivery of healthcare, but also in terms of the impact financially related to this market and to this procedure. So the Warrior Watch project had two intentions. Number one, is divided, what we call a Warrior Watch infection study. And it's really to use these physiological metric as well as the psychological clinical assessment to understand the effect of the infection on people. And can we use these measures to try to detect the disease differently? And the other one is more related to what you're interested in here is the Warrior Watch Mind study, and trying to decipher the psychological impact of COVID on our healthcare workers across all occupation using these, again, mobile health solutions. And the mobile health solution that we have adopted, and this is really thanks to knowledge and work that we have at Mount Sinai, especially with the Hassel-Plattner Mount Sinai Institute and the group that is expert in working with application and adopting these apps from your different platforms and different wearables to try to detect the disease. So we've worked and specifically in here using the Apple Watch. This is the one I'm wearing also here, to use the Apple Watch to get real-time 24-hour assessment of physiological measures that include, as you know very well, heart rate, and we'll focus a lot on heart rate activity variability, but then other things being also measured. But I'll show you why the heart rate variability is an important measure here for us. And we marry the watch with the Apple Health app that we can access and create and modify a specific application of the Warrior Watch, where we could get subject to a journal to us what is the clinical symptoms that they're getting, and specifically here, what are the COVID-19 symptoms, but also more importantly is the aspect of psychological well-being with different instruments and that we can put via this app in a very friendly and I would say, doesn't take a lot of time to do, and you will see that we've had a very high level of compliance. So the surveys that we ask will be related to the infection as mentioned, but let me also stress the aspect of the surveys we use for resilience, stress, optimism, emotional support, quality of life, and other physical markers of stress on the body. So the heart rate variability is one of the measures that we focus on. It really measures the activity of the autonomous nervous system. Our body continuously readjusts to maintain this balance. So actually heart rate variability is important, and it's actually essential for that balance. And it obviously it is something that has an effect on the autonomous nervous system, on the heart rate, monitoring or modulating heart rate, blood pressure, glucose, etc., to keep everything in balance. And this is a very important marker to try to assess basically disease, but also health. And it relates to this interaction of our autonomous nervous system, of both the sympathetic nervous system and the parasympathetic nervous system. Okay, so let's start first about maybe just to give you a sense of this program. Let's start with the infection prediction, although here our focus is not, for this symposium, is not necessarily the infection, but this is a good example to show you, how our story evolved from looking at the infection all the way to the aspect of stress, which we'll finish with. So again, the platform can do both. We can get this assessment by patient, but also measure this heart rate variability in real time. All this, it's important to talk about this, deploying this contactless, especially during the pandemic. So you're able to continue to do measurements on subject, on a patient, without having to see them face to face, because that creates obviously difficulties, obviously from an operational point of view, but also from a safety point of view also. So in this part of the study, the part one related to infection, we've enrolled over 300 patients from April to September of 2020, and the median age of these healthcare workers are 36, 70% women, and about 7% reported having a positive COVID PCR test prior. We measured and conducted this with a 42 days follow-up. And one of the things that's important to point out here is using this technology, we noticed that, you know, compared to other studies or other ways of collecting this kind of data, that we had a very high sustained participation, over 70% in the study. So that, I think, is a good lesson in terms of deploying these tools and the importance of having compliance and making sure you have high compliance so you can have a very well performed study. But let me talk to you a little bit about the infection detection and how we looked at this. We looked at this over over a period of somebody looking at the daily heart rate variability on days with COVID positive and COVID negative diagnosis, and being able to look at one of the parameters. There are a lot of ways you could measure, or sorry, you could process the heart rate variability. I'm not going to dive a lot into the detail, but just to show you here on the, let me see if I can get my pointer to cooperate. Not easily. On the left-hand side, you can see the significant differences in heart rate variability, both pattern and amplitude in the two groups who are COVID positive or negative. And we can see now when we break it down into the COVID negative healthy, we're trying to see, are we able to look at this daily patterns over time to see if we can detect different signatures, prior or after the COVID positive. So you can see now, if we look at the healthy, a very nice waveform pattern, high amplitude, specific signature in terms of what how it looks like. But now when you have somebody is 1 week before the COVID positive test, now we have major changes in both the pattern but the amplitude. It's really obvious, and you don't need much sophisticated person to see that red curve now compared to the green curve is very different. But now if we look at now longitudinally, 1 week after the COVID positive test, the waveform continues to be different, but starting to get the shape. When we see now 2 weeks after, now it's starting to gain similar to the healthy, but it's a different amplitude, but the pattern is there. So you can see that these patterns or the amplitude of the signal is very much related to the COVID infection, or any infection, I would say, in general. And it's very much, in a way, important. It's important that we're able to detect it much before somebody is symptomatic. So you can see here, this is a very intriguing way of potentially finding infection in general, and using that information to maybe isolate that subject, just so you can err on the side of caution. Very simple, so you don't have to get that swab, and could again be important managing the infection during the pandemic. Okay, so now to get to the meat here that you're interested in, which is the aspect of the psychological well-being, again, using the same platform that I just discussed with you, related to heart rate variability measurement by the watch, as well as the surveys that are done by the subject in a very friendly manner, and deployed via the app on the phone. So our goal here, just to restate it, was to understand the psychological impact of COVID, and specifically looking at vulnerable patient group, or subject group, I should say, which is the healthcare workers. And this was done really across our Mount Sinai health system and across all occupations. This is too much detail, but just to tell you in terms of the instrument that we've used, these are well-established and validated instrument. We've used most of the time the short, I will say, version of these instruments, the resilience scale, the life orientation test, as well as the general health and the quality of life. Again, all programmed in a nice way and can be done very fast. I was one of the subjects that also participated into the study. I was able to do this on a daily basis. It didn't take too much of my time, and I was, tried to be, and I was compliant because, again, it was not too onerous in terms of effort to be able to do this. And these are important things when you are designing mobile health applications or testing, to be able to make sure that the technology you're using is not asking too much from you, especially if you want to collect data on a daily basis, but also over a longer period of time. So in terms of about our cohort, about 360 in this case, we have distribution between staff, but also in terms of clinical non-trainee as well as clinical trainee. And you'll see this was important to be able to divide that, because you'll see there are some trends in terms of these different healthcare workers groups. And so one of the things, in terms of baseline, the clinical trainees had higher baseline resilience versus on the CD risk-2 test versus the clinical non-trainees as well as staff. There was higher optimism also in these clinical trainees by looking at the life orientation test and emotional support versus the staff. And also the clinical trainees had also higher emotional support compared to the clinical non-trainees. Now, looking at the longitudinal factors associated with the, factors associated with longitudinal stress, we see that there is increased longitudinal stress as the number of the COVID-19 cases rise, as well as we've also saw increased longitudinal stress when there was 4 weeks before a COVID-19 diagnosis. Decreased longitudinal stress happened in people with baseline resilience, with baseline emotional support, and baseline quality of life. So all these were important factors in terms of that stress, perceived stress on the subject. And occupation and COVID-19 related work exposure was not associated with the long-term stress. So what have we learned now in terms of looking at the change in perceived stress? So we've seen change in stress after COVID-19 diagnosis, with decreased only in people with high emotional support or high resilience. Very interesting, obviously, finding. And change in stress related to the COVID-19 case number was tied together only in those with high emotional support or high resilience. I'm not gonna, yeah. Now, looking at the physiological stress analysis, and again, relying on this heart rate variability measure, we see again these, as we've seen in the infection, there's significant reduction in heart rate variability between participants here, in this case, with high versus medium and low emotional support. And then we've also seen, significant changes in the circadian pattern of heart rate variability when the cohort was stratified based on the baseline resilience. So these really, finding, demonstrate to us things that we've known, I think, or in the literature has been known, but really measured here uniquely, and also in a unique setting, that demonstrate that low resilience, emotional support, and quality of life, can identify healthcare worker at risk of perceived and physiological longitudinal stress secondary to, let's say, a COVID-19 pandemic. And then this assessment of healthcare worker for these features can help, obviously, permit an allocation, of psychological support. And you will hear, from Dr. Marin about how Mount Sinai is doing this, but also looking at, isolating or figuring out who is at risk during the pandemic, and really redeploying also these healthcare workers to try to manage this. Then I'll finish with you with a study that is a third study of this program, of the Warrior Watch Health. We talked about the infection, we talked about the mind, but also now we are launching what we call the Warrior Shield. And the Warrior Shield is focused on trying to see if we can build resilience by using one of the established tools, which relates to biofeedback. The tool specifically is called HeartMath, and it's been shown to improve patient or employee satisfaction in organizations, as well as being effective in dealing with anxiety and depression, and also building resilience. It's been tried in the many healthcare system settings in the past. Just to give you a little bit what it is, this is in the area of biofeedback. It's also in a mobile health platform. You have a wearable sensor that you attach to the subject earlobe, and then you have an app with some training that helps you look at your heart rate, and with some practice sessions related to managing stress, but also managing some breathing, etc. You reach, and you learn how to reach that state of coherence, and affect, obviously, in a positive way, your heart rate variability, but also, as shown in the previous studies, shown that how you could manage and build resilience. So the Warrior Watch Shield, study is launching, soon. We are waiting and pending IRB approval, and we plan to test this in our cohort of this 500 healthcare workers that we evaluated before in a 5-week intervention. And we hope to report on that very soon. So with this, I finish with, again, all the mentors that in the field of psychiatry, psychology, including James Moreau, Dennis Charney, and others, but also my partner in crime, really takes credit here, Robert Hurton, who is a GI doc at Mount Sinai that I met through the COVID pandemic. We were what we call the COVID kids, getting together, learning about what they're doing in their field of research, and trying to see how we could synergize by building these mobile health application to look at these interesting parameters. So thank you again for your interest.
Thank you so much, Zahi. That was great. And I don't think I overpromised that this is really a revolutionary way to follow people and have direct clinical implications. So thanks so much for that. There are two questions that came up for the Q&A. I think we have time to address those. One by Evelina and one by Lana. If you could read them out and answer them, that would be great. Yeah,
let me get to that thing. So the Evelina is the first question. Thank you. Okay, so the question is, are you able to analyze disturbance in circadian rhythm? So no, we have not looked at that yet, but we are interested in circadian rhythm in general, and I think it's obviously related very much to the aspect of stress and managing stressors in general. So that would be of interest to us, and we are certainly looking at other sensors, including Apple Watch, but also other sensors to be able to monitor that. The other question is about the effect of quarantine without infection. We did not look at that, but that easily can be done. And part of the surveys, you could look at that. Lana's question, Lana, as these become more popular, I wonder what protocol risks are in place for people or for person who indicate severe psychological symptoms and or those who indicate suicidal ideation. I'm going to leave this to Dr. Marin. I mean, Dr. Marin is, with her program, is putting together ways to manage this and to deal with this and building all different tools. So I hope that this mobile technology, again, is a window to people's mind through this physiological measures that again help us, with management of these subjects. So we're very excited to learn again from this group and to continue this collaboration. Thank you.
All right, Zahir, thanks so much. And again, it was a great talk, and you made a great segue to the next and last speaker of this symposium, Dr. Deborah Marin, who will introduce, what we really do, for our own people. So how are we gonna help our own people? So Dr. Marin is the director of this newly established Mount Sinai Center for Stress, Resilience, and Personal Growth, which was initiated at the peak of the pandemic, let's say last summer, because we all realized that we need to help our own. And this was an initiative, I think, initiated by the Dean, Dennis Charney, and has been supported by philanthropy and other funds. So Dr. Marin received her MD and completed her residency in psychiatry at Mount Sinai, followed by a research fellowship in mood disorders at Cornell, and then she returned to Mount Sinai in 1992. Since then, she had had several leadership positions, including Chief of Geriatric Psychiatry, Dean for Clinical Research, and Chief Medical Officer. She has been the principal investigator in NIH-funded grants that have investigated the clinical and biological correlates of normal aging and dementia. And her clinical interests include treatment of memory disorders and depression. And as said, she is now leading this program, which I think is a great initiative and has very important implications, obviously for our own, but also hopefully will be followed out throughout the country and serve as a model how we can help our own cope with this severe crisis that's still ongoing. So please take it away. Looking forward to your talk.
So basically, I want to talk about the fact that, we had to turn on a dime. You heard a lot about what was being done at Mount Sinai, so from a research perspective. But, basically March 11th was the benchmark of our system changing rapidly over a period of days. And within a period of weeks, we had to convert our existing service lines into basically COVID hospitals. We have a system and they were all COVID hospitals. And staff were not only reassigned to units, but their basic communities were disrupted because they were taken away from their teams, and they were also asked to do things they would never had to do before. Mortality rate was so high that in one of our hospitals, by the end of March, they had hit the yearly rate of deaths in 2019. Visitation was not permitted, so patients were isolated. And as was mentioned before, we didn't know, proning was even thought about. We didn't know what medications to use. And the fear of infection on the part of the workers was very high. Just to show you, this is actually the atrium of our hospital. This is where Starbucks is. This was converted into a nursing unit, and pods were built over the course of 7 days to accommodate what was thought to be overflow of patients. And this hospital has 1,100 beds. This shows actually what it looked like for patients. Everything was done actually to minimize contact between the healthcare workers and the patient for fear of infection, so that the work was done outside of the room. And healthcare workers also then had to become the conduit, if you will, between the patient and their family, since no family was allowed. And our buildings looks different. These holes are inpatient rooms for negative pressure rooms for folks on ventilation. So, we live in a hospital and medical school where both the dean, and the CEO are psychiatrists. And you heard a lot about Dennis Charney and his work on trauma and resilience. So in the period of basically 5 work days, a multidisciplinary task force was set up for the immediate response. And that included several departments, which I'll talk about in a bit. And we actually initiated a proactive outreach to highest need employees, which were pretty much everywhere. But the emergency departments and the ICUs were particularly hard hit. And also mental health liaisons were deployed to these areas to walk around to get a pulse of what was going on. In addition, everything was virtual, but different psychosocial support was provided for employees, including groups, individual counseling, guided self-care activities, and these wonderful recharge rooms were developed in the clinical areas where folks worked, where they could actually take a time to take a break. And we are a system of 42,000 employees. And one of the hardest things was actually how to get people to know where to go to get resources. And that took a lot of collaboration with the Office of Wellbeing and Resilience under John Ripp, Department of Psychiatry, Social Work, and Employee Assistance. You heard about the expected stress-related conditions. I do want to talk about one thing here. I'm not talking much about moral injury, but that's something that in our clinical practice, we're hearing more about how folks really are feeling that they should have, could have done something different, and moral injury can lead to stress-related conditions. And of course, the issue of burnout has to be considered as well. So this study actually is the study that was done at Mount Sinai by John Ripp and his group to look at what happened to our healthcare workers. And the results are exactly what you saw for other places, but this was very close and personal for us, that this was done. The survey was sent out to over 6,000 folks and a pretty good response rate of 55.8%. And the sample analyzed was 2,579. And what was found was that 39% of healthcare workers did screen in for either a mood disorder, major depression, an anxiety disorder, or a post-traumatic stress disorder. I'm only showing this slide, which actually was developed by Samsha to understand the fact that this is not something that comes and goes. There was a lot of heroic behavior, and a lot of the healthcare workers, by the way, did not like being called heroes, just for you to know. So it was commonly used, but not necessarily well received. The problem is that there are multiple ways of the pandemic. There are multiple other things going on in addition, to COVID-19, and we clearly are in this disillusionment phase. We're trying very hard to address it, but this is not an acute response to an acute situation. It has to be an ongoing response. So toward that end, it was decided actually in April that a center had to be developed that could manage long-term the issues related to the situation for our healthcare workers. And fortunately, there was a lot of experience, as you already heard, in trauma and resilience and program development. And here, the issue here is that there was 18 years of experience at Mount Sinai in the World Trade Center program for first responders. And in addition, and actually 3 of the 4 of us who really started the center, Dr. Katz, Dr. Piero, and Dr. Sharma, were involved, if not led, actually that program. And Dr. Katz actually had global experience in disaster psychiatry with tremendous leadership support, including from the board. And fortunately, we had a lot of relationships with other folks prior to doing this, which really facilitated kind of a rapid deployment of a center. We were basically asked to go live in June, and we started thinking about developing this in April. We had a lot of data to prove that there was a need, and we had philanthropy, which is very helpful. The challenge was that each hospital has its own culture. Coordination of efforts with multiple departments was an issue. Getting the word out to all stakeholders we knew would be an issue. The people were very busy. We had approval from Human Resources that people could take breaks to come and do workshops with us, but there was a shortage of staff. That was very hard, and that led to how we decided to have to deploy a center. We have 13 unions at Mount Sinai. We have multiple insurance coverages. It was the decision on senior leadership that we were not going to charge employees for the care. So we had to have a heavy lift with multiple carriers to get a no cost sharing done. And also there's the issue of getting over the stigma. So we developed the center. It is a centralized resource for all faculty and staff. Everybody in the health system has access to us. The goal was to develop resilience training workshops, develop an app, and focus on community engagement. And was decided very early on that we had to have our own treatment program, since in the psychiatry behavioral health clinics and their faculty practices were already 90% occupancy and getting a tremendous amount of need from the community that we serve. But this center at this time is only for folks who either work or learn, in our health system. So the first thing that was done was actually Craig Katz took the chapters from the book that Dr. Charney and Southwick wrote and developed 10 workshops based upon the 10 factors. What we learned early on was that people were very interested in doing this, but people couldn't attend. it was very hard to get folks to come to so many workshops. So they were reduced down to 5 workshops. One is on faith, meaning, and spirituality. Another on social support and role models. Another facing fears, active coping, self-care, and realistic optimism. And these basically are educational sessions where participants learn about the resilience factor. They learn how to actually adapt something and discuss creating a plan for themselves, which is actually being something that we're looking at now in an app that's being developed. And actually 99 workshops have now been conducted with a wide variety of healthcare workers. It does turn out that residents actually are more likely, or physicians, because they're more likely to be able to carve out time for didactics. But we are working with nursing, physician assistants, and other support staff. So this is the Wellness Hub mobile app. It was initially deployed, and thank you, Zahi, actually, to get us in contact with the Hasso-Plattner Institute for Digital Health. Because basically in a month, they built an app for us. And the app was actually first to be a confidential screening device with well-established scales regarding both issues of stress-related conditions, but also well-being and also spirituality. And the app now is on to more of a V3, if you will, where basically this is an example, the landing page. So one is learn about factors of resilience. The other one is journaling how one is doing, both related to resilience and day-to-day activities. My progress and getting support. My progress, folks can log in to these different scales that assess either mood, anxiety, depression, and well-being, as I mentioned. And based upon their score, they immediately get a feedback. Green means you're doing well. Yellow means the cutoff score is above what we expected, and people then get prompts of ways that they can reach us or other folks for support. What now is in the app are actually videos about each of the factors of resilience. And this version of the app is focusing on actually now making this a resilience promoting opportunity for individuals to engage with. Each person who clicks in to Introduction to Resilience and one of the workshops we just talked about, they basically learn about it and then discuss it and then develop a plan of action. And based upon activities, they're now badges to support them. In addition, folks now have the ability, actually from the beginning, to get to know us immediately, call us, email us, and other activities that can help with them. My calmer activities could be yoga, could be music, mental health support, and chaplaincy. The chaplains are very much integrated with this center, actually, in terms of rounding on staff. They're very well integrated into the clinical teams at Mount Sinai. So getting the word out. We adopted what's called the accelerator model for community-based participatory research, which has been done at our institute here in health equity and population health. The idea here was to get stakeholders who, from different disciplines and different hospitals, and hear what they think about what we're doing. And we meet on a monthly basis. They were the ones who actually said from the very beginning, you can't just do iOS, you have to have this in Android. Anything you do has to be bilingual. You can't just have flyers in English. They gave us very good ideas about how to get the word out, because a lot of the folks who work at Mount Sinai don't have easy access to a computer, and blast emails don't work. So we developed flyers, we developed 10 cards. We have staff who around in the hospitals. We have chocolate, thousands of chocolate bars, 10,000 coffee sleeves to get the word out with a QR code to get to us too. So clinical care. It is available for all staff, trainees, students, and faculty. And we have arranged at no cost sharing to participants that they have up to 14 visits with no out-of-pocket cost for them. There's, of course, pharmacotherapy, and we have trained social workers and psychologists, Dr. Piero, psychiatrist, Dr. Katz, who actually also perform cognitive behavioral therapy, interpersonal therapy. And we opened our doors to the center, as I said, June 20th. We opened the doors for the workshops July. We opened our clinical program in October. And as you can see, we started small, 31 visits in October. It doubled in November. It doubled again in January, and it keeps on growing. And actually, due to staff turnover and vacations, we don't have as large a size as we would think of patient visits. But the folks who are coming here are very symptomatic. 14% of suicidal ideation, for example. And we know we're only hitting the tip of the iceberg. We have over 1,000 visits, and over 100 patients treated, but it's definitely people are suffering, and we hope we can get more folks to learn about us and come to us, and we're doing all that outreach toward that end. This could not have been accomplished without a lot of partnerships with Office of Wellbeing Resilience, Hazel Platinum Institute, Psychiatry, Human Resources, Population Health, Office of Diversity Inclusion, Nursing, Social Work, Support Services, GME, and Rehabilitation Department. I bring it up because in order to get something like this going, you need so much help from so many folks, and we were lucky actually, to get that help. This is who we are. As I mentioned, Dr. Sharma, DePiero, and Katz are all psychiatrists who all worked in World Trade Center or ran the program here. We have a chaplain who does our community engagement. We have a staff psychiatrist, Dr. Vivalakwa, program manager, our social workers, another chaplain, and administrative support. So with that, I thank you all, and I'm sorry about the screen share being the way it is. I hope I got the point across.
All right, Debbie, you definitely did. I think it worked out fine. So thanks very much. And, this is a great initiative, and you've brought it to fruition very quickly. I mean, it's really, really going very well with lots of, people actually getting help, and the numbers are growing as we speak almost. So there is a question from Jill Barron. I mean, you want to pick that up? And there's
Sure.
another question. So Deb, if you just read them out and answer them, please.
Is the wellness app available for purchase for non-Mount Sinai employees? If so, what's its name? Thank you. It's not available for purchase. Actually, it's all employees can download it for free from a central repository here. It's anonymous, but right now it's an inward-facing app. But thank you. We plan on it being an outward-facing app, but we actually are embarking on a randomized controlled trial to see how it does work actually promoting resilience. It's called the Wellness Hub. The next question, what's the name of the app? It's called the Wellness Hub app.
All right. Yeah. I mean, it would be nice though if people will follow this lead, right? If we get other people who actually are interested either to purchase or to use it for free or whatever. You think that's going to be happening in the foreseeable future? We're going to make this available.
There are a lot of apps out there that purport, to do a lot of things, but there's no data to prove that they do what they say they're going to do. So we're being very cautious and methodical. And James Murrow, by the way, thank you for helping us a lot. I just said that you're ready. He's mentoring us on a lot of these things. We really want to test whether or not this app really does promote resilience. And I think that it's a very necessary mandatory step before we start vending it anywhere.
Yeah, that's a very good point. One more question by Eddie Golden.
Wellness Hub new version will be available to Mount Sinai Health System by Science Central. Thank you, Eddie. Yes, Eddie's amazing. She and her team actually developed this app for us, remarkable. All
right, great. Okay, thank you very much, Deb. That was great. If there are other questions, actually, there are actually two other issues. You want to pick those up?
Cynthia Green. Hi, Cynthia. There are other populations similar in need of clinical assessment and resilience training that would benefit from this app, such as those in senior living. Will be interesting for comparison purposes too. There's no question that we have talked about, and there is a great app actually Dr. Hurton was involved in developing for folks with actually irritable bowel disease. So one way is to think about tailoring the app based upon the population that it may serve, be it a chronic illness, be it certain folks who are vulnerable. So there's no question something like that would be very useful. Matthew Dobbs, terrific resources and outreach initiatives. Did any patient needs arise as you developed the Wellness Hub that you didn't initially expect to surprise you? In terms of the app use, I think that it's not a treatment tool. It's just an assessment tool at this point resilience promoting. I think the real issue is, what do we find with the folks who we treated? And what we found was that the whole era of we're going through now is so stressful that it's not just about treating COVID-19 patients, but that we're hearing just a lot of stress and anxiety in general on our healthcare workers.
Okay, and then the final one is a thank you, which is great, and which is, I think, a great way to end. Thank you all very much. The speakers was a great symposium. Was with a very nice overview of what we can do to identify the people at risk, and even develop models, as Scott Russo has shown, develop new treatments, as James has shown, develop new apps and systems to work, as Zahi has shown, and then finally implement everything, as Deb has shown. So thank you very much. I really enjoyed having you here. Thank you also all the delegates, all the attendees who participated in this virtual conference. Thanks all, stay well, and all the very best. Bye-bye.